today's talk is actually by the research group of Professor Neelam Sinha. And today we have with us Professor Jaya Sreevalsan Nair as our session chair. So Professor Jaya is currently an associate professor here at IIIT-Bangalore. She leads the graphics visualization computing lab here at IIITB. And she's also the core member of the eHealth Research Center, EHRC, at IIITB. So without further delay, let me hand over the stage to Professor Jaya. So as always, you can type your questions on the chat boxes, both on YouTube as well as Zoom. And also after the talk, there will be a dedicated Q&A session. Thank you. Over to you, Professor Jaya.
Thank you, Professor Nikhil. So I'd like to take this opportunity to introduce the talk and the speakers for the day. So as Nikhil already mentioned, this is the last talk in the medical AI series, and it's quite exciting to have 4 speakers talking on medical imaging data of different dimensionality. So today the talk will cover one-dimensional, two-dimensional, up to 3 and 4-dimensional data. And each speaker will speak about their work in each of for each of these data sets. So the talk is titled Medical Image and Signal Analysis, and the talk is given by 4 PhD students from 3 PhD students and 1 postdoctoral researcher. This is from the lab of Professor Neelam Sinha. So today we have with us Dr. Gopika Gopan, who is currently a postdoctoral researcher at IIIT Bangalore. She obtained her PhD from IIITB in 2020 on her thesis, EEG Time Series Classification and Analysis. And she was one of the recipients of the Best PhD Thesis Award at our convocation for her work on her thesis. Then we have with us Mr. Sunil Kumar Vengalil, who is a PhD scholar currently. He is a PhD candidate currently, and he's working on real-world and medical image analysis, deep learning-based approaches. Mr. Chakka Sai Pradeep is a PhD scholar working on surgical video analysis, and he focuses on adapting deep learning-based approaches for providing visual surgical assistance. Then we finally have Ms. Snigdha Agarwal, who is also a PhD scholar with us. She's working on trauma CT scan analysis, and she's working in collaboration with doctors at NIMHANS for the trauma assistance. So we are very excited to have this talk today. So without much ado, I will pass on the baton to Dr. Gopika. Thank you.
Thank you, ma'am. I hope my screen is visible. Today's talk will be on medical image and signal analysis work under Professor Neelam Sinha. There will be 4 presenters today. So the work under this consists of data ranging from signal, image to videos. Currently, the work consists of EEG time series analysis, fundal image analysis, surgical video analysis, CT and MRI scan analysis. In terms of EEG analysis, the current words includes baseline detection, distinguishing state of mental arithmetic and rest, distinguishing negative and positive emotion, and distinguishing subjects with Parkinson's from normal. In terms of fundal image analysis, the objective is to detect optic disc, segment blood vessels, and microaneurysms. In surgical video analysis, the aim is to identify and track the surgical tools and hence the phase of surgery. In CT analysis, there are 2 distinct work being carried out currently. One is COVID screening using CT scans, and second is trauma analysis, where we detect areas of trauma in brain CT. In terms of MRI analysis, we have 2 works. One is MRI reconstruction using deep learning approach. The other is tumor analysis, where we detect and segment tumors. So there are 4 talks in this session. The first talk consists of EEG time series analysis, followed by fundal image analysis and surgical video analysis, with concluding talk on CT scan analysis. So let's move on to part 1, which is EEG time series analysis. So for this session, we'll be talking on analysis of EEG signals for visual creativity assessment. This work also had collaboration from an IIM Tech, SVR Aditya Reddy, and also Professor Madhav Rao. So the outline of the work is as follows. We'll introduce the EEG creativity, the innovation, the device use, task description, data collection, proposed methodology, followed by analysis, simulation results. So creativity can be considered as an important trait found in humans and can be viewed as a cognitive basis of a conscious thought. So the studies involving EEG in creativity started in 1975, where they started studying the correlation of cortical responses and creativity. Now, multiple studies have explored the correlation between the alpha band in EEG in visual creativity, verbal creativity, and creative ideation. In terms of EEG creativity, we are focusing on visual creativity tasks, and there are two studies on which our present work is focused on. The first is the study by Nagornova, and second is the study by Jalon. In the first study, they tried to assess the change in EEG during creativity with the condition that all the drawings has to be carried out with a given set of geometric shape. So two creative tasks are present. The first is drawing any original picture, and second is drawing an original picture of an object as indicated. In terms of control tasks, there are two. One is recreating a picture that is shown prior to the participants, and second is drawing the geometric shape from a set continuously. Now, they have observed that the created tasks contain higher EEG power. In the second study, they tried to assess the change of creativity level when copying or when a person is creating one's own drawing or a text description. So here there are two tasks. The first is in creative tasks, the first is writing a description of the word displayed, and second is drawing a picture to represent the displayed word. In control tasks, the two of them are copying a given text and copying a given drawing. So what they observed was that the control tasks were more automatic and is related to motor response, while the creative tasks resulted in brain waves that were associated with higher cognitive process. So the inference is that when a person is doing an original drawing or an original text, there is higher creativity involved. With this in mind, two tasks were designed. One is most creative task, that is drawing a landscape, and second is less creative task, that is repeated drawing of given geometric patterns. So the basic innovation introduced is one, assessing grades of creativity using EEG for novel task design, that is creativity involving originality, creativity while copying a given shape, and a monotonous task. We have introduced a new monotonous task over here, that is repeated tally mark, and also using a commercially available single channel EEG headset for analysis of visual creativity. So the hypothesis that we are basing is that the EEG contains characteristic signatures pertaining to the phenomenon of visual creativity, and we can exploit this to quantify and assess the varying grades of creativity with the design task. So the device used is Neurostein Mindwave device, which is just a single channel device. We are obtaining the data from FP1, that is frontopolar region of the prefrontal cortex. It contains an inbuilt noise and artifact removal algorithm, and it has a sampling frequency of 512 Hertz. So the major tasks, there are four main tasks over there. The first task is the max creative task, where the subject is asked to draw or illustrate a landscape with the condition that it must have a mountain, a coconut tree, a hut, and a river. A sample is given on the left side, and they are encouraged to add more details, and no prior planning is given. The next one is a less creative task, where they had to repeatedly draw the given geometric pattern. The third is a nil creative task, where they have to repeatedly draw the tally marks for the given time, that is 5 minutes. And finally, we have a rest, where the subjects can sit comfortably in the chair with closed eyes. So this is the timing sequence. We can observe that each one of them had 5 minutes of the task. The row B shows an example of the subject's drawing, and C show the corresponding EEG obtained. So the data were collected from 18 subjects of our own IIITB students of the age group 20 to 28. They are not formally trained artists, and we had equal number of males and females. So at the beginning of the experiment, they had to answer a questionnaire which consisted of rating themselves as an artist, describing their emotional state at the start of the experiment, and also elaborating a comedy scene or meme just to lighten their mood. So the whole experiment was for 30 minutes with an intertask interval of 1 minute. Now, this work was not funded by any organization, and due consent was obtained from the subjects, and it was carried out in accordance with Declaration of Helsinki. So the proposed methodology contained 4 different classification framework. Initially, the EEG from neurosky device underwent pre-processing, that is normalization, artifact removal. Then for statistical and chaos-based analysis, wavelet decomposition was carried out. Now the 3 off-the-shelf classifier, that is SVM, Ensemble, AdaBoost, and KNN were utilized in terms of source distribution, statistical analysis, and chaos-based analysis. So in pre-processing, initially the single channel time series were segregated into different classes based on the start and end time, and few overlap samples were removed. Now, an additional artifact removal algorithm based on wavelet was utilized in case there are some residual artifacts that are not removed by the neurosky built-in artifact removal algorithm. The filtered signals were then normalized. In case of statistical and chaos analysis, the signal further underwent 6-level wavelet decomposition, of which we use DB4 mother wavelet. So in terms of chaos analysis, our main aim is to capture the change in nonlinear dynamics of the brain. So we have used off-the-shelf features over here, Hurst exponent to estimate the self-similarity, detrended fluctuation analysis to estimate the amount of long-term correlation of the EEG, correlation dimension to estimate the complexity of the brain, the approximate entropy to estimate the complexity of the signal, largest Lyapunov exponent to estimate the chaoticity change in the system, and CATS fractal dimension to assess the fractal patterns. In terms of statistical features, what we intend to do is understand the statistical characteristics in EEG signals using energy, which sort of indicate the activation of the brain region, entropy, the amount of irregularity or uncertainty in EEG, mean and variance, followed by skewness, kurtosis, which capture the different characteristics of the distribution, median absolute deviation, and interquartile range captures the information related to statistical variability and statistical dispersion, followed by Joth mobility and complexity, which capture the characteristic relevant to the trace of the EEG. Now, the source distribution analysis, we have utilized moment generating function as a feature. So this moment generating function is calculated using the equation shown over here, where each of the MK are the corresponding kth moment calculated as shown in the equation. Now, the T is a value in the real space. Over here, we have taken it as ranging from 190 to 200. In terms of Lstm analysis, we used a stacked Lstm with the base Lstm layer shown on the left side. And we had 4 Lstm layers, each with a dimension of 100, followed by 2 dense layer and a softmax layer. We used stochastic gradient descent optimization, and learning rate was 0.001 with a dropout of 20%. So the simulation results obtained from chaos analysis are shown over here. In particular, if we look at different grades of analysis of visual creativity, we see that there is significant changes in the chaoticity of the beta band. However, when we add the rest into the different grades of creativity, it's seen that all the features are significant in distinguishing the 4 classes, and we reach a peak accuracy of 52%. This is a confusion matrix, and it can be seen that the rest is correctly classified completely, and there is no misclassification, while the misclassification is more related to the grades of visual creativity. And the other performance metrics show that the obtained results are significant. In terms of statistical analysis, we see that for both the grades of creativity as well as when you include the rest, Joth mobility and complexity is the feature that is significant. That means there is significant changes in the characteristics of the trace of the EEG signal to distinguish the different classes. So over here, we obtain 38%. However, we see that the rest is actually misclassified to the nil creative class, while there is no other misclassification to the other 2 classes for the rest. In terms of distribution analysis, overall, if you look, the performance is less compared to the previous 2 analysis. However, we were still able to obtain 38% accuracy similar to that of the statistical analysis. And over here in the confusion matrix, we see that the rest is actually misclassified to both least creative and nil creative, which was not the case previously. And we were able to obtain 38% accuracy. On the right side, we see a sample of the 4 tasks moment generating function versus the T value. And we have taken the T value for our analysis from 190 to 200, where we actually see significant differences between the MGF of the 4 tasks. Now, the simulation result of LSTM is shown over here. We can observe that the rest and the least creative are completely misclassified. So one of the possible reason is that we have very limited data. It's just 18 subjects. So the LSTM is unable to capture the characteristics that is required for distinguishing between them. Even with this misclassification, it was still able to obtain 38%, which was similar to that of the statistical as well as source-based analysis. Now, there are a lot of limitations for this pilot study. First thing is that this study is based on subjects who didn't have any disorder. So if you include the subjects where there are disorders present, there will be changes in the nonlinear dynamics. However, the question will be whether these changes will be aggravated by the medical condition, or would these medical condition introduce additional changes. The second drawback is that it's a pretty limited data. It's just 18 subjects with no diversity in age group. So if you want to extend the study into a generalized age group or across generalized age group, then we need more data collection and also analysis. Third is that the tasks introduced were pretty coarse visual creativity, and it do not capture the complete finer grades of visual creativity. And hence, with the addition of more subjects of varied medical condition and artistic talent, the study would be more comprehensive. So overall, the major objective of this work was to classify tasks with varying levels of creativity using off-the-shelf EEG headset, that is NeuroSky Mindwave. So we carried out statistical source distribution, chaos, and LSTM-based analysis, and it was found that for the 4-class classification, where the chance accuracy is 25%, the peak accuracy obtained was 52% in terms of chaos-based analysis, indicating that the changes in nonlinear dynamics of the brain are involved when we are distinguishing different grades or different coarse grades of visual creativity. Also, it shows that the captured EEG does contain characteristics pertaining to difference in grades of creativity. So what this work intended to give is a generalized framework with limitation that can be extended using additional data and different modalities for further analysis, including monitoring changes in cognitive dynamics. At present, there is a work that is being carried out and additional work with multi-channel EEG, which are in initial phases of data collection. So this basically concludes the EEG part of it. Now let's move on to the part 2 of it, which is fundal image analysis. Over to you, Sunil.
So this work was done in the Machine Intelligence and Robotics Center, which is supported by Government of Karnataka. So here we developed a deep learning based tool for automatic screening of fundal images. Okay. Basically, fundal images are images of eye. So what you see here is the simple cross section of eye, human eye. So our human eye is a 3-layer structure with the retina in the innermost layer is the retina, and where all the photosensors are arranged, and the middle layer called choroid is the one which is spread with a lot of blood vessels, and that gives the enough oxygen to all the retinal cells. And the final layer, which is shown in black color here, is the one that holds retina together with the it's actually act as a protective measure for the inner eye. Now, some of the important structures we are interested in is one is optical disc. So here you can see a bundle of optic nerves leaving the eye. So that region is called optic disc. And second important region that is of our important interest is called macula. So this is the region where the light is focused in the retina, and this region is packed with a lot of cone cells, and hence it is responsible for the focused vision part of art. So we will be this is the fundal image. So fundal imaging is an image of retina is taken using a special camera called fundal camera, and this image shows various structures. Like first and most important structure that we are interested in is optic disc, which is the the brightest region, which is highlighted over here. And another region is macula, which is the dark region, which is highlighted. So this corresponds to the macula. And third visible structure that is shown in fundal image is blood vessels. Okay. So our aim is to segment out these structures in a fundal image, the blood vessels, macula, and optic disc, doing a segmentation. And in addition to this, so what we saw earlier was the image of a healthy person. Now, when there are various diseases or abnormalities present, you can see some lesions here. What is shown in the white patches here, the lesions are called exudates. So these exudates are mostly seen in diabetic retinopathy people, patients of diabetic retinopathy. This is the fluids leaking out of the blood vessels. Okay. Now, other anomaly that is seen in visible in fundal image is called hemorrhages, which is bleeding from the retina. So here the red patches, which is highlighted over here, is the hemorrhages. Okay, this is another example of hemorrhages. Now, another very tiny, this is the most challenging part, I would say, is to segment in a fundal image. So these are called micro aneurysms, which is happening due to small opening of the capillary walls. And these are in dimension 100 to 10 to 100 micrometers. For this study, for training, we used some of the publicly available data sets. So we use images of data sets of different resolutions, ranging from the drive data set, which is of lowest resolution of 565 cross 584, to the HRF, which is high resolution fundal images, and then even higher resolutions, which is IDRID. IDRID stands for Indian Diabetic Retinopathy Image Database. So these are images of Indian patients with various levels of diabetic retinopathy identified. So each of these data sets comes with the different types of ground truths. So they also have mixture of images with healthy and different diseases. Okay. So as I was telling them, in this project, mainly we are interested in segmenting out the regions like blood vessels, optic disc, fovea. Fovea is another term for macula, and exudates. So we use deep neural network with multitasking for segmenting these structures. Okay. So when we use multitasking, the correlation between different structures are exploited. So we use this is the structure of the network that we use. So this structure of network is called a UNet, and this is a very famous network used for medical image segmentation. So this has multiple layers of convolutional and deconvolutional layers. So the path on the left side is called the encoder, where the input image is fed, and the successive layers of convolution and max pooling layers decrease the size of the image. And then finally, on the right side, what path is known as a decoder, and the decoder path takes the image, and then it consists of deconvolution layers, which actually upsample. So each deconvolution layer will upsample the image, and finally at the output you get the same image mask of the same resolution as the input image. So in the output we have two different channels for predicting two different structures. So in this example, for example, the channel shown on the top one gives a binary output image, which is the predicted optic disc, and the one below gives you a binary output image, which is predicted blood vessels. Now we'll go over to see some of the results of these experiments. So these are some of the results on high resolution fundal image, HRF images. So what you see in the green in the top row, the third image plotted in the green is the prediction for the blood vessels, and the middle one is a ground truth. So all the green pixels over here are the correctly predicted blood vessel pixels, and whereas the small red spots that you see here is the missed ones. So those blood vessels, the model were unable to detect those. So as you can see, most of them are very tiny blood vessels, which are very fine structures that the model has missed out. And the below one, the bottom one that you see is the optic disc original image, ground truth, and the rightmost one is a prediction. Okay, again using the same color convention. Now, these are some of the results on IDRID dataset for exudates and fovea detection. So, as you could see, the prediction on the exudates also has some very tiny missing pixels, and this we are actually trying to improve on these results. And the below one is a fovea prediction. And so, in fact, we tried out different experiments with the different types of training losses. So what we observed is that when we use dice loss, when we use a combination of both dice loss and binary cross entropy loss, we obtained a better result. So this is probably because when the dice loss, actually gradients will become very small when the number of predicted targets are very less. So initial convergence will be faster once you use binary cross entropy loss, and once it is converged, then the dice loss can directly optimize the target. Okay, and so these are some of the numerical results metrics that we got for blood vessel segmentation. So on blood vessel segmentation using combination of dice loss and BCE loss, we were able to get a dice score of 82% on HRF dataset, which is a high resolution dataset. And the lowest one you can see 66.37 is on STAIR dataset. And this dataset is very obsolete now, and the image quality in these datasets are very poor. This is the oldest dataset for the Fundal image, STAIR. Now, same for exudates, we got a F1 score or dice score of 60%. This is still work in progress and is being improved actually. And for optic disk, we got a very good F1 score of 94%. Yeah, and I would also like to give you a small 3-minute video of our tool. So we developed a tool which is running a web-based tool where someone can upload a image and make some of the predictions and see the results on that. Okay, so I'll just so one can upload one's own image over here, Fundal image, and yeah. So here I'm selecting an image just downloaded from internet, and the tool will go on the prediction. So initially we check the quality of the uploaded image and we report the quality score. So this one is a very good image with a quality score of 0.91. A quality score of 0 is a poor and 1 is the best quality score. And now I can select what all kind of analysis I need to perform. Like here I want to do blood vessel and optical segmentation. The tool is currently integrated only with these two. Other things are still needs to be done. And I can get the predictions over these. Okay, so this is the blood vessel prediction, and what you see below is the optic disc prediction. Now I can optionally upload the ground truth also. If I have the ground truth images, I can upload the ground truths also, whereby I can actually get a comparison and I can compute all the metrics with the prediction and ground truth. So here I'm just uploading the ground truth for blood vessel and optic disc. This is the optic disc ground truth I'm uploading. Okay, so this is the metrics that is computed. For this particular image, I was able to get a F1 score of 51%, and for blood vessels and 90% for OD, optic disc. And now I can also see, so here only the prediction is separately shown in two different images. I can show the both prediction and ground truth in one single image with a color coding scheme. If I select the combined option. So here again, the view shows the green pixels shows the correctly predicted one. Red pixels shows the missing one, the pixels which is missing, and the blue one shows the extra predicted. If a blood vessel is not there and it is predicted, it is shown in blue color. Okay. Yeah, I think that's all. And with this, I will hand over to Pradeep, who will be talking about the surgical video analysis in the next session. Thanks.
Good afternoon, everyone. I'll be coming to the part 3 of the today's session. It is on surgical video analysis. So today we'll be looking at specifically on the endoscopic perspective, and we will try to analyze those videos and provide some kind of assistance to the surgeon. In this regard, our work is we have done a multitasking. We have proposed a multitasking DASD architecture for laparoscopic cholecystectomy surgical assistance systems. This work was done at Machine Intelligence lab at IIIT-Bangalore. This work also has been published in ISB 2022. The outline of this particular section of this today's talk would be, we'll start off with a sample use case, that is the cholecystectomy, and then we'll move on to the introduction, then data set information. We'll look at the existing literature. We'll look at the proposed methodology to some extent, and the training methodology results, and finally the conclusion. So what is cholecystectomy? Cholecystectomy is the process of removing of gallbladder from the abdomen. So few of the medical conditions would force some of the patients to get their gallbladder removed. Right? Typically, the process, the surgery involves removal of gallbladder. So this gallbladder is connected to the body in three parts. So the gallbladder is connected to the liver with a tissue, and the blood supply is coming from cystic artery, and the gallbladder which contains the bile juice is provided to the stomach through a cystic duct. So we need to sever the connections of these. If you remove these three connections, and we will be able to package this gallbladder and remove it out of the body. So widely accepted surgical phases for this particular surgery are 7. So the first one would be preparation stage, then the callet triangle dissection, then clipping and cutting, gallbladder dissection, gallbladder packaging, cleaning and coagulation, and gallbladder retraction. This surgery is typically performed using these 7 tools. Grasper, used for holding different body parts inside, bipolar, hook, scissors, clipper, irrigator, and specimen bag. Specimen bag is to towards the end of the surgery, if we would be placing this gallbladder inside the specimen bag, and during the gallbladder retraction stage, we'll be pulling out the specimen bag. Irrigator is to either pour in some distilled water and cleaning for the cleaning purpose. So it'll both pour in water and suck out water as well, right? And the clipper will be used for clipping parts of the duct or cystic artery, so that the flow can be stopped, and then we can cut it out. So the cutting process is done by scissors. Hook is used for cutting out different parts. To separate gallbladder and liver, we need to cut out the tissue. So the tissue part is removed with the help of hook. Even the callet's triangle is also filled with contains a lot of tissue. So that tissue part also is removed with the help of hook. The bipolar is used for so if there is some blood leakage inside the body, inside during the surgery. So that's kind of a stitching operation is done with bipolar with the help of electricity. So it will just burn the skin so that there will not be any bleeding coming out. Grasper is used to hold different parts of the organ at places. Yeah, this is one sample video of the laparoscopic cholecystectomy. This is how it would look like. This is clipping and cutting phase with the help of scissors. This is scissors, and this is the grasper. This is the gallbladder. So we have cut out the cystic duct and the cystic artery. So these two are the cystic duct and arteries. You can see these are the clips. With the help of scissors, we have cut out the. Yeah, so clips are already placed. So this is one small snippet of the surgery. Right. So this is how a typical surgery looks like. The vision. This is just to show how a doctor would look inside the patient. So this is an endoscopic perspective. Coming to the introduction would be so we have talked most of it. So our surgical tool presence detection, surgical tool localization, and phase classification are an important aspect in providing additional surgical visual, and also in determining surgeon skills. So so whole intention is to provide assistance to the surgeon, right? So for this particular task, that is the simultaneous tool localization, tool presence, and phase classification. In cholecystectomy surgery, we have used these 3 data sets: M2Chi16 tool location data set. So this data set is provided by Stanford University. Serena Young team has provided the labels for this particular data set. M2Chi16 tool and Colic80 data sets are provided by University Hospital of Strasbourg. So Colic80 data set is a parent directory, a parent data set, out of which M2Chi16 tool and M2Chi16 tool location data sets are subsets. Right. So this particular data set will provide only the tool presence. That means each frame has a particular tool in it or not. And M2Chi16 tool location data set provides the bounding boxes of few of the images. So each one is a subset of the other. This is a data distribution Venn diagram. Coming to the data set information used in our task, we utilize a data split of 80-20. That is, we got 2025 images for training and 506 images for testing. This is provided by M2Chi16 tool location dataset. Colic80 and M2Chi16 tool datasets were used exclusively for evaluation. M2Chi16 location dataset provides only the class of the surgical tool and its bounding box coordinates. For our training, we have borrowed the corresponding labels of surgical phase and surgical tool presence from Colic80 dataset for these images. So these are the results coming out of our model. So if you look at this example, this is a surgical phase in the top left, and this is the bounding box for the tool. So this is a gallbladder dissection performed by hook. When we look at something else, yeah, cleaning and coagulation performed by bipolar. So the bipolar tool, and the surgical phase detected is cleaning and coagulation. Coming to the literature related to this work, temporal convolutions were used in Techno paper for the first time for surgical phase classification. Authors had used causal dilated multi-stage temporal convolution networks in their work, which had achieved the then current state of the art results. However, tool localization and tool presence detections tasks were not performed in this method. Multitask learning was used on phase classification and tool presence detection in EndoNet and MTRSNet. However, tool localization task was not performed in these methods. To the best of our knowledge, our proposed method is the first method addressing surgical tool presence detection, tool localization, and phase classification in a multitasking manner. So coming to our proposed methodology, so this is we propose a multitasking deconvolutional single shot detector, in short MTDSD architecture, with resonant 101 backbone. Right. So these are the three tasks in hand which we are targeting. So surgical tool localization, this provides the bounding boxes along with the tool name. So bounding box of the tool and the corresponding what is the tool present inside the bounding box, surgical phase, and surgical tool presence detection. Right. For the task of tool localization, we use features from different layers which are carrying varying receptive fields for predicting spatial presence of various surgical tools. So then we have an encoder decoder hourglass architecture with feature maps being carried forward. This addresses the challenge posed by shrinking feature map size and lower receptive fields effectively. Our method leverages on the strong correlation between cholecystectomy surgical phases and the specific surgical tools usage. Initially, we have the dataset is provided. So this is how a typical inference would look like. Input video would be converted to frames of size 3x320 x 320, and this would be passed into this backbone architecture. And this would extract important features and pass them to the SSD layers. SSD layers and the DSSD layers together would provide us with feature maps of various scales. So the importance of this feature maps of various scales is that we can identify or we can detect objects of different sizes. So that is the significance of usage of this various sized feature maps. And finally, non-max suppression is used to decide on the most prominent bounding box. contribution of the study is to design a multitasking architecture to perform surgical tool presence detection, tool localization, and phase classification simultaneously. Also, we leverage on the deconvolutional single shot detector architecture to perform surgical tool localization. We also achieve state-of-the-art accuracies on publicly available M2K16 tool location datasets in a multitasking manner with fewer parameters. So this is a proposed combined loss function. So we use a combination loss of that is cross entropy softmax loss for surgical phase classification, a multi-label soft margin 1 versus all loss function for surgical tool presence detection, spatial surgical tool classification. This is first proposed in single shot detector paper. And surgical tool bounding box regression loss function. This is also part of single shot detector paper. Right. So we propose the usage of multitask learning framework. Along with tool localization, tool identification, we provide additional supervision in the form of surgical phase and surgical tool presence at the end of encoder. This part. So you also, this is the end of the encoder. So here with we add additional convolution and FC layers for and provide labels of surgical phase for surgical phase classification. And another layers of convolution and FC, parallel layer of convolution and FC layers for surgical tool presence detection. Right. Model was trained jointly over the stated 3 objectives. Coming to the results, here we show surgical tool usage frequency versus surgical tool phase on M2K16 tool dataset over surgical videos of 14 and 15. The first row, when you look at the first column, will give us predictions coming out of a network, and the second column will give us the ground truths. This is corresponding to the surgery 14. So this is a full length surgery has been evaluated to see to check how our model was performing. Also to analyze which particular tool is being used at which particular phase. So this correlation also has been exploited in the training phase. These two particular plots of the first plot is corresponding to surgical phase classification results versus ground truth time progression. These are also on M2K16 tool dataset or surgical videos 14 and 15. So, for illustration purpose, we have used only surgical videos 14 and 15. Right. First row is corresponding to prediction, and second row is corresponding to the ground truth. Coming to the next part of the results, surgical tool presence detection results versus ground truth time progression. Here again, it's a time sequence bar. First part is related to the predictions, and second part is related to the ground. Is related. These A to G are corresponding to different tools. Then coming to the numbers, accuracy numbers, surgical tool localization results on M2K16 tool location dataset is shown here. We benchmark the performance of the popular generic object detection methodologies on M2K16 tool location dataset. To the best of our knowledge, this is the first attempt to create such a benchmark on this dataset. We obtained 20% improvement in terms of MAP with respect to the available state-of-the-art methodologies. Here we show surgical phase classification results on Colec 80 dataset. Techno, which is the state-of-the-art methodology, it performs a single task of surgical phase classification using resident 50 backbone and temporal convolution network, which this has reported 88% accuracy. Our proposed method trained over just 2025 images achieves 80% accuracy on the same Colec 80 dataset. So this particular methodology had used 2.3 million images. We have only used 2025 images. Coming to the surgical tool presence detection, we report an accuracy of 97.51% on M2K16 tool location dataset. State-of-the-art methodologies are trained on Colec 80 or M2K16 tool datasets. These benchmark are on different datasets. These each of them provide 2.3 million on training and 0.6 million images for evaluation. Our proposed method trained over just 2025 images achieves 65.44% accuracy on M2K16 tool dataset. Coming to the conclusion, a surgical phase-aware multitasking encoder-decoder DSD architecture for surgical tool presence detection, tool localization, and phase classification and laparoscopic cholecystectomy surgery was proposed. Correlation between surgical phases and tools was exploited by usage of additional supervision in the form of surgical phases and tool presence at the end of encoder to improve on the surgical tool localization. The proposed multitasking approach achieved MAP of 91.9% for surgical tool localization. This is 20% higher than state-of-the-art methodologies. 97.77% accuracy for phase classification, 97.51% MAP for tool presence detection on publicly available laparoscopic cholecystectomy surgery dataset. Same 3 tasks, when performed individually with similar architectures, would require 72.6% higher parameters. So we have achieved higher accuracies with lesser parameters. So this would enable us to port this methodology or the POC onto edge devices, and we'll be able to achieve real-time results out of it. Yeah, thank you. So coming to the part 4, it is on the subject intracranial hemorrhage detection. Over to you, Snigdha.
Thank you, Pradeep. I hope everybody can see my screen and hear me well.
Yes.
So today I'm going to present part 4 of this session, which is intracranial hemorrhage detection. So in this, what I'm going to present today is an end-to-end application for intracranial hemorrhage detection using CT scans. This is an ongoing work which is being done in collaboration with the Department of Neurology in NIMHANS in the guidance of Dr. Jitendra Saini and Professor Neelam Sinha. So I would talk to you about the outline. So I would talk about the problem of traumatic brain injury, focusing on types of hemorrhage, then the data set that we're working on, the project workflow, the first task, which is to identify normal and abnormal scans. Then I would go on to show some of the results of a first task. Then the second task is to build a UI-based annotation tool, which we are currently working on, and the next steps. So traumatic brain injury is a major issue which covers 1/4 of global deaths in India. Traumatic brain injury is increasingly being recognized as a public health problem, and it is of most concern in middle-income countries. In India, more than 1 lakh lives are lost every year, with over 1 million people dying from these injuries. In India, 1 out of 6 trauma victims die, while in the United States, this figure is 1 out of 200. So it's very important for us for the appropriate management of TBI for survival of these patients. According to NIMHANS, there are a major part of these CT scans, traumatic brain injury scans, that go unreported. So our task is to report these scans at the earliest, so that some of the lives from these trauma injuries can be saved. Types of hemorrhages. So there are 5 types of hemorrhages that we are targeting. The first one is intraparenchymal hemorrhage. Intraparenchymal hemorrhage happens inside of the brain. So as you can see here, this blob, which is the blood oozing out of any artery or a vein, this usually happens with a high blood pressure. In some cases, intraparenchymal hemorrhage can also be caused due to trauma, but those cases are less. The next one is the intraventricular hemorrhage. This hemorrhage happens inside of the ventricular. This shape that you see here, the butterfly kind of shape, this here is the intraventricular hemorrhage. The subarachnoid hemorrhage happens between the arachnoid and the pia mater. So usually it's like it gets spread on, the blood gets spread on like most parts, and sometimes in between as well. This type of hemorrhage happens due to trauma in some cases, but again is not a very common in trauma. It usually happens with the rupture of aneurysm. The most common type of hemorrhage is the subdural hemorrhage. This hemorrhage happens between the dura and the arachnoid. So basically, the outer part, which is the skull, and then in the middle of the soft portion. So this is where the subdural hemorrhage happens, and is the most common cause of hemorrhage which happens in trauma cases. And most of the patients with subdural hemorrhage, 50% of them go into coma as soon as they incur this. So it's very important for us to identify a subdural hemorrhage. The other type of hemorrhage is epidural hemorrhage, which happens again between the dura and the skull, but that's outside of the extreme outside portion of the skull and the dura. Epidural hemorrhage usually is followed by a fracture. So in most cases where the skull cracks, we would see an epidural hemorrhage. The reason for epidural hemorrhage is also a major reason is trauma, because a skull fracture will happen in cases of accidents, but epidural hemorrhage is seen very less in the real world data. So it's like very less data is present for epidural hemorrhages. The data set that we are talking about today is we are solving this problem using an open source data set, which is the Rsna data set. So this is an open source data set which contains CT scans already segregated into images. So CT scan is separated into images. There are a total of 7 lakh images spanning across 25,000 patients. This problem is a multi-label classification problem where each patient can also have more than one type of hemorrhage. The normal images that we have in this data set are about 6 lakh, and the abnormal images are about 1 lakh. So here on the right side, you can see the spread of the images. Intraparenchymal, subdural, intraventricular, subarachnoid, and epidural. As you can see, the epidural hemorrhage is very less in real world data because it's not a very common case. But the subdural hemorrhage happens is the maximum occurring in the real data set. The second data set that we have is from NIMHANS. So we are currently at the initial evaluation using the NIMHANS data set. We are currently targeting a normal images of 100 and abnormal images 100 for this. We are going to span this to a future labeling task where we would be labeling CT scans from 2,000 patients. So that's the target that we have. The project workflow is in this format. So we initially target to build a model which is able to identify normal and abnormal scans, so that from the data set that we have in NIMHANS, we could segregate the normal scans and the abnormal scans. After this, we use this model to evaluate the efficacy from this RSNA model on a small data set of the NIMHANS data. We extend this model to identify all types of hemorrhages in a multi-label scenario. On top of this, we plan to build a UI-based tool to perform ground truth labeling of all types of hemorrhages, hemorrhages which the radiologists would label in the NIMHANS data set. We extend this tool to further classify all types of hemorrhages and segment the area of the hemorrhage on the larger labeled NIMHANS data. So the first task that we picked up was to identify a normal and an abnormal scan. So we worked with only the subdural hemorrhage type as the normal class for now, as according to the physician, this was the dominant class in the NIMHANS CT and was of most concern to be reported. So for this, from the RSNA data set, we have used the abnormal 47,000 images, and we have randomly sampled from all the normal images 50,000 images. The initial results that we have are from three off-the-shelf models. So these architectures are picked from this RSNA data set. The people who have worked on it, the literature survey that we did. So people have tried on the ResNet, the EfficientNet. The Connext architecture, which is the latest one, is something that we have tried for our data set. So with ResNet 18, we were at an AUROC of 0.64. With EfficientNet, we have an AUROC of 0.79, and with Connext, we have an AUROC of 0.84. These are some of the results from our abnormal modeling. So the predicted category here in the first image, as you can see, we have predicted as abnormal. The actual class is abnormal. The probability is 0.80 for the abnormal class. This is only for subdural images. So as you can see that the artifact of the hemorrhage is here. The next image also is pretty clear with the abnormal and the abnormal subdural portion. The predicted, the third image, we have predicted it as normal, but the actual is an abnormal image. The reason that we see why this would be happening is because this is a multi labeling. The intraparenchymal and subdural were the two hemorrhages in this data. So as I had said before, there are images where we see more than one hemorrhage class. So here, since the intraparenchymal data was not provided to the model, it was not able to do a good job. The other one, the final one is again a misclassification. So this also we have predicted as normal, but the actual is abnormal, where the majority portion is a subarachnoid hemorrhage and a smaller portion is a subdural hemorrhage. So we couldn't identify the hemorrhage in this case. We are working on improving this model further. So the task 2 that we are currently targeting is a UI-based annotation tool. So for the radiologists at NIMHANS, it is important for them to have a tool which can do labeling, where they can just click and select the class of the scan that they are looking at. So so what we have identified is that on the right side we have the scan opened for the radiologist, and on the left side we have all the kinds of labels that we want to add: normal, intraparenchymal, intraventricular, subarachnoid, subdural, epidural, and so on which the radiologist can very quickly click, and a CSV file can be generated, which gives us the ground truth labeling for the multiple scans that we are targeting right now. So this is a very huge labeling that we are looking at. So a tool like this would assist the radiologists in labeling the data set faster. So the next steps that we're targeting are to work on the UI tool to assist to right now check our model based on the 100 abnormal scans which we are labeling manually, and evaluate the initial model on the NIMHANS dataset. And basis this, we would also study what is the difference between these huge data sets which are available as open source, and if they are really of worth in real world scenario. So these are the steps that we're working on. This was all for today's session. We are now open for any questions from anyone. Thank you.
So thank you, Snigdha. Thank you, all the other speakers as well. So now the floor is open for questions. You can maybe unmute and ask. It's a small crowd. So, any questions for the 4 speakers?
This is for Sunil. So are floaters also detected as blood vessels by falsely detected as blood vessels, or how is it?
I'm sorry, can you repeat the question? What is falsely detected as blood vessel?
No, floaters in the eye, are they detected when they're doing the image detection, or how is it?
No, actually,
So Sunil,
that
the
is not
floaters
detecting.
don't show up, right?
Yeah, that is not shown coming at all. So
that doesn't show up in the fundal
in the fundal image
I
at
see.
all. Yes.
Okay.
Yes.
Okay. Thanks.
Yeah. Thanks, Ashok. Any other questions? Okay, so while the other questions are coming in, I'll ask a few. So I'll go to Gopika first. So Gopika, regarding your work, it's fantastic work. So I wanted to check a couple of things. One is, I mean, it's more like I didn't understand and I need more clarity sort of thing. So in terms of the ground truth, what do you say, generation for the data that you have in terms of creativity levels for your images, how did you go about that? Like, do you have an expert evaluating them?
Not exactly. So what we did was we asked them, like this was actually conducted early in the morning in one of the labs. So when we are giving them a task, there are two of us over there. One will be monitoring the given EEG and all that. The other person will be actually monitoring the subject to ensure that they are actually concentrated on the work that is being carried out.
Sure.
So there is no expert as such. It's like we manually ourselves, we made sure that there is no any kind of disruption that is being occurred over there. So like we are not giving any kind of pre-planning. For example, in max creative tasks, when we ask them to do, we say, okay, you have to draw a landscape with these conditions. Now start. And immediately they have to start. We note down the time. And one of us will monitor that they are actually continuously doing the work. Anytime any disruption occurs, immediately we note down the time so that we can remove those part where the disruption has actually occurred.
Okay. So your task itself was labeled as most creative, least creative, and
Yes.
medium
Yeah.
creative. Okay. My next question is regarding. So you said you want to increase the number of channels in
Yes.
the measurements that you take through the EEG measurements, right? Observations. So,
Yes.
what is the hypothesis that you have, like going from 1 to 16 or
Okay,
whatever?
so the main hypothesis is that we actually want to use commercially available EEG headset. We don't want any medical, so that anyone can actually utilize the headset and obtain the EEG for further analysis. So the basic one that we started off is this Neurosky, which is a single channel. The next set that we are actually currently being used in association with Mindro is Inobio headset, which consists of 8 channels. So the idea is that even though creativity, we are able to obtain some amount of characteristics in our FP1, that is over here, we were able to obtain some amount of information. It's not just a single lobe that is being carried out. So there is also occipital lobe, temporal lobe, some amount of changes are present there also. So using just a single channel, we just want to see how far along we can obtain this kind of characteristics for distinction. But now we want to extend it to cover both the prefrontal part, frontal part. I think there is a central part and 2 parietal part also. So together we want to cover the entire thing and see now how well we can actually obtain the distinction between the grades.
Okay. So will you need a baseline observation which is done by the medical professionals, because they would be experts, right? In terms of your evaluation. So then you can go ahead and probably say with confidence that 8 channel is working as close as
Yes. So actually, before going that, we want to set up the basic framework for it. So this was actually a pilot study. This was more a pilot study, then extended it to see how well we can include. So apart from the 8 channel, we are also including a difference in data. So earlier it was just our students. Now it is our students plus students of architecture. So they are more inclined to the drawing aspect than us. So once we include that and we get, okay, at least to some extent a good performance, then we were thinking we'll include some medical like expertise also into this to see how much more we can expand. So these were just the pilot study that we actually started up. It's not the final thing. So the final thing, it requires a lot of modifications, a lot of incorporation of different kinds of data, especially when we try to include the disorder kind of data. So these are just normal people data.
Thank you, Gopika. Now I'll go to Sunil. Sunil, I wanted to ask you, so you had this nice image of green color, which is true positive, and red, which is false positive sort of thing.
Right.
So did you have any sort of understanding on if the size of the blood vessel itself, like the thin versus thick blood vessels, did it help you in analyzing your data? Like, did it give any kind of characteristic results?
Yeah. So one observation, as I was showing, what we've seen is that most of the one that is missed out are very thin ones. So what we are doing now as a part of to improve that, because all the results what I showed today was after resizing the images. So the image resolution, these are high resolution images. but we had to, for the to fit into the memory of neural network, we had to reduce the size. So we reduced it to 512 cross 512. So we are hoping that if we process the image at the original resolution, we might be able to detect even the smaller ones. So what we are doing is we are doing it patchwise now. So instead of, because blood vessel is spread across the entire thing, and so you can create patch, and each patch can be given to the neural network. So in that case, you are doing it at original resolution. So that we are seeing some improvement, but I haven't shown those results in this today's this thing. That is a new thing. Yes, yeah.
Okay, so then your learning is also done on the patch-wise images, and
Yeah.
then you can.
Yes.
Okay.
Yes. Yeah.
So then it will learn on that, and okay,
Yes.
yeah. Because the image showed that you're missing out on the thinner blood vessels.
Exactly. Yes.
So yeah. So are these blood vessels significant in the first place? I mean, biologically, is it something that
Yes.
the doctors look at? Or,
Yes. So one place where they look at is like these certain topological characteristics of these blood vessels, like tortuosity and all, how much curvature is there. So that depends, evaluated for many cardiovascular diseases, they say. So that gives you a correlation with cardiovascular diseases. And second thing is, if you can use this, so these features also, it can improve other detection like diabetic retinopathy and all, right? So for detecting the anomalies, so first if you segment out and uh, keep aside the normal structures, then you can look for the anomalies like exudates and hemorrhages and all. So yeah, that was the plan.
So what you're saying is even the thin blood vessels are significant for medical diagnosis and prognosis. And
yes.
Thank you so much.
Yeah, thank you.
Now I'll go to Pradeep. Yeah, so Pradeep, I got confused with one of your slides with the results. You said this is the first time the benchmark task is done, but you still had a comparison with other state of the art. I think that was for the tool localization.
Yes, yes.
How did you compare with the state of the art if this is the first time? I mean,
This,
I think I missed what you were saying there.
I'm sharing my slide.
Yes, please go ahead.
Yes.
Yeah, your second point, to the best of our knowledge, this is the first attempt.
Yeah. So when it comes to surgical tool localization, there is no benchmark as such. So these are popular object detection methodologies, so which I have trained on, and so these are the methodologies used for ADAS, normally Advanced Driver Assistance System. So they use for object detection algorithm. So I have ran this particular available data sets or the labels which I have in hand on the other most popular object detection algorithms and our proposed method with the which was trained on multiple.
Okay, while you're on your slide deck, there was another slide on the usage of the tools. This one. So you the surgery 14 and surgery 15, right? So the predicted one is highly fragmented. The top images, the surgical phase classification. So how do you explain that for your method? And I mean, this clearly needs to be improved also, right?
So when the ground truth was labeled by the surgeons, so they did not really worry what was the action happening in between during this course of the surgery. So when there are some tools involved which are not supposed to be present in that particular phase, so there is no hard and fast rule they had put. So they put it some not very hard rules, but soft rules they had put. So they just labeled like, so when as this first phase starts, right, preparation stage, so there is no possibility that any other phase is happening during that in reality, even if there is some small fraction of small seconds of other phase happening over there, they won't really label it. So it is just phase wise labeling and in sequential order. So when I have trained the network, I did not want to enforce that sequential thing into the prediction, because so they didn't take the effort to label each and every frame, so they just labeled it the time frames alone. So but I didn't want to spoil it, so I left it. I avoided the sequential information part. So I could have included that in the training, but I did not. So if I had included that in the training, then this would not happen. So I believe this is more appropriate.
So when you say there is surgical phase classification, this is not necessarily a phase corresponds to a single tool, right? So you can have multiple tools used in a particular phase.
So for example,
what are the features that get captured there?
so for example, what happens is during the phase of cleaning and coagulation, so these on the x-axis it is the phases over here, and this is the relation between the tools. So so these are bars of the tools, right? So so cleaning and coagulation, there will be a lot of interchanging of tools or actual work happening between gallbladder packaging, cleaning and coagulation, and gallbladder traction, or even sometimes gallbladder dissection. All of this while during the process of gallbladder dissection as well, if there is a small cut, there is a possibility of cleaning and coagulation happening for a few seconds inside this phase itself. So, but those labels are not available in the ground truth.
Okay.
So there is and there is a correlation between the tools, because what I mean was if I'm performing a color triangle dissection is basically cutting. So there most probably this can be only be done with the help of a hook. Hook will actually cutting. When looking at clipping and cutting, most probably this is happening with scissors and with help of clippers, right? Yeah. So there's a direct relation between phase and tools used. So so no other methodology had actually used that in correlation. So, but I had provided into the network both the labels from with different data sets, of course, on this available on the same image. So that is how I achieved the improvement here.
You seem to know all the surgical tools very well. Maybe you can pursue brain surgery as well at some point.
Yeah, I've been looking at the data set for too long. Maybe I know that only the names of it, not thing.
Right. Interesting. Thank you, Pradeep. I'll ask Snigdha a question.
Yeah, sure.
So, Snigdha, yeah, here. So, I had a question on the scans that you showed. So, do you? So, for each patient, you will have several scans, right? I mean, you're looking at certain slices alone, right? So, how do you figure out which is the optimal slice to use for a particular trauma? Like, do you have to look at the size of the injury or something? I mean, is that something that you do as a pre-processing step, or is there?
So yeah, so initially, like each scan, we are taking each slice as an image. So that's the portion. So with that, like each slice, we know that it contains a hemorrhage or not. So that segregation is already happened when we segregate that. But we also do windowing on each of these images, because the radiologist also see only a portion of where the artifact or the hemorrhage is present. So to make that hemorrhage intensified, we do a set of pre-processing step, a set of windowing step, which enhances the portions of the blood, which as you would have probably seen, I can share my screen again, from the images. The image so this color, we have tried to change the way this looks to make these brighter. Otherwise they're more like in this, the images are in this fashion, which are a lot of noise, and so this is a bigger hemorrhage, so it's very clear. But cases where the hemorrhage is very subtle, it's very difficult to identify, so that windowing helps. But each scan is separated slice wise on if it contains a hemorrhage or not.
Okay, so for a single patient, you can send multiple slices for learning. I mean, whichever slice has shown hemorrhage and you have enhanced it using your image processing methods, you would use it for the. So in your learning models, do you use the color images or the black and white images?
Black and white images only, ma'am.
So you converted back to grayscale. Oh, that was this illustration book.
Yes, that's like just for illustration. And most like in all the CT scans, like in most CT scans, we can also see like a different, like more than one hemorrhage type also. So one hemorrhage type might be very visible in one slice, but some other hemorrhage will only be visible on like the start position of the head and the edge. So that is the reason, like in some slices, the model doesn't perform well. Like this here, parenchymal is very visible, but subdural was lesser one. But maybe another image of this scan would show the subdural hemorrhage more. So that's where the model confuses. Yeah.
So for each slice, do you give a probability of a certain hemorrhage type, or I mean, as in, so because you said it's a multi-label classification, right? So it is deterministic or probabilistic the way you present the results?
Right, ma'am. So currently we have only built this model on the subdural class, and that is the reason why where the cases where we have a combination we are not able to do well because we have not shown the model all the data. So right now it is only one class, so we are getting the probability of one class. But when we extend this to all the 5 hemorrhage types, then we would be evaluating it on a probability of each class.
Okay. So in terms of what you're going to do, will it make sense to have 5 different classifiers for the different types of hemorrhage, or do you want to have just one which will learn for all types, which would be better?
Yes, ma'am. So with the current progress that this area has made, having 5 models is an overkill. So one model should be able to identify all the 5 classes. But we have seen that the epidural class, where there's very less data, doesn't really perform well. So maybe that can be a separate model, and we can work on an ensemble strategy there. But otherwise, having 5 separate models, considering the literature that is already the work that has already been done in architectures in deep learning methodology, having 5 architectures would be an overkill initially.
Makes sense. So thank you, Snigdha. Yeah.
Thank you.
I think we are done. I don't see any other questions. And so with that, I think I'd like to thank all the speakers and wonderful work that they're doing at IIITB. So with that, I'll pass on the baton back to Nikhil. So thank you all. Bye.
Thank you, Jaya, and all the speakers. So with this, we are actually concluding our ongoing series on medical AI. And next week, we'll be back with a new series, which is a new topic, which we have named as Vistas in Spatial Temporal Modeling.